I'm Monica with Nucleic Verse. I use analogies, metaphors, and anthropomorphization in order to create poems and story. It's how I understand. It's how I see the unseen and ponder the unknown. Just as words are imperfect models of thought, so too are these words strung together as story. But imperfect as they are, we try. Today, I have a poem and a metaphor. So strap in and hold on. First, the poem. Central Dogma. Sheltered is the original. In the museum, catching a glimpse of its reflection as it is resealed after secret extraction. This half version of itself is enough, has been enough, will be enough, can slip through the century, relay the ribosome process 3 for 1, base for acid. Ghostly reflection leaves the factory, prize protein emergent, take shape and form. Structure is function. It is all for this. Let's discuss first a metaphor, and then let's dissect the whole thing together, mixing the poem and the metaphor up, but let's see if this word salad works. This is the metaphor. What can a cookbook library teach us about the central dogma of cell biology? What even is the central dogma of cell biology? Stick a pin in the second question. Let's just learn about the Doris Kirshner Cookbook Library at the University of Minnesota, St. Paul campus first. The dealio is you can review any of the 5,600 items which contain recipes bound in pages of books and pamphlets. You cannot remove any of the items from the library. So how is a crafty little cook get away with making a recipe from this collection? Luckily, I had a tool and post-it note. I wrote down all of the information needed, and now I could take the super duper useful post-it note outside of the library. Alrighty, back to our pinned question, and we'll mix in the poem and the metaphor to try and get a better understanding of central dogma. What is the central dogma of cell biology? The central dogma is this. DNA goes to make RNA, goes to make a protein. Just to review in the metaphor, we went to the library, selected a recipe, copied it down, and then we went out and made our delicious casserole. So we're seeing that there's 3 steps, and also we describe 3 steps in the poem. DNA is used to make RNA, is used to make a protein. Just like in our library recipe, DNA, the cookbook can't go out of a region of the cell, the nucleus. So a copy is made that retains the critical information and can go out of the nucleus. That's the RNA. This RNA Post-it note, and I think that's so cool because Post-it notes are sticky on one side and so is the RNA. I'm easily pleased. Can be used to assemble proteins at the ribosome. The ribosome is kind of like the cook. Why does the cell do this? Double-stranded DNA is sturdier than a single strand of RNA, and we want to protect it inside the nucleus, and we don't want it going willy-nilly. We want it right there and organized in a way that we can access the recipes anytime we need them, just like the cookbooks in the library. RNA is single-stranded and it's destined to break down. It can go out of the nucleus and into the cytoplasm to find the ribosome. The ribosome can translate 3 bases into 1 amino acid and start making a chain that can fold into a structure that has a function. Every 3 bases of RNA direct the ribosome chef to add another amino acid to the growing string. This string can fold and be crafted into multiple shapes, which perform different functions in the cell. These proteins are so cool. They can do anything. Break molecules apart, help bring them together, escort molecules across all sorts of membrane structures, or they are pores in membranes themselves. These are the tools that get everything in the cell done. Let's read the poem again and see if it makes more sense to us. Central Dogma. Sheltered is the original in the museum, catching a glimpse of its reflection as it is resealed after secret extraction. This half version is enough, has been enough, will be enough, can slip through sentry, relay to the ribosome, process 3 for 1, base for acid. Ghostly reflection leaves the factory, cries protein emergent, takes shape and form. Structure is function. It is all for this. So let's take a look at some of the words here. Sheltered is the original. This is the DNA in the nucleus, and it is like a museum. You can see the actual remnants of evolution. Catching a glimpse of its reflection, imagine an RNA polymerase opening up a double-stranded DNA to transcribe a section of it and having the RNA fragment, the transcript, floating out of it, and then having the DNA resealed, reannealed together to be double-stranded again. That's what I was thinking about when I wrote that. And this half version, this RNA transcript, is only single-stranded, but it is enough. It carries enough information. and it can pass through the nucleic pores, the centries, and relay to the ribosome the information from the DNA that it will never touch. It relays this information 3 bases, and those 3 bases can be translated into a protein. Every 3 bases is 1 amino acid. The ghostly reflection refers to the messenger RNA that is leaving the ribosomal factory. And the prize protein is prized because it is the reason why all of that happened. It is the end goal. As it emerges from the ribosome, it happens quite fast, and it folds as it does so according to the amino acids that are being linked together. And it takes that shape that instructs its function. And again, you will hear in cell biology, molecular genetics, everything molecular, that structure is function. If the structure changes, the function changes. And that's as far as I'm going to get into opening that can of worms, because it's a big one. But we have a lot more to talk about as far as that is concerned. So thank you.